Hi, my name is Stephanie Paris, and today I'm going to discuss the mutation in gene regulation. What is mutation? Mutation occurs when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. Here are some examples of mutations in plants, animals, and humans. A mutation in this garden mouse roses has caused some of the flowers to grow orange rather than yellow. A fairly common mutation in mice causes bunds in the coat around the body. A common mutation in humans causes red blood cells to be shaped in a sickle pattern. Mutations can be inherited, and also mutations can be germline mutation. Germline mutation is any detectable variation within germ cells. And mutation also can be de novo mutation. De novo mutation is a genetic alteration that is present for the first time in one family member as a result of a variant in a germ cell of one of the parents. And mutations can be also acquired mutation. Acquired mutations is a genetic change that occurs in a single cell after the conception of an individual. Types of mutations. First is the substitutions. A substitution mutation is a type of replication error during DNA replication which places the wrong nucleotide or sequence of nucleotides in the wrong position. Substitutions is important of currently used materials. With less hazardous materials is one of the most effective ways of eliminating or reducing exposure to material that are toxic or pose other hazards. And the next one is insertion. Insertion means exactly what you would expect. An extra nucleotide is added into a DNA sequence. Insertion mutation can be small, involving a single extra DNA base pair, or large, involving a piece of a chromosome. Deletion. A deletion mutation is a mistake in the DNA replication process which removes nucleotides from the genome. A deletion mutation can remove a single nucleotide or entire sequences of nucleotides. And the next one is a frame shift. A frame shift mutation is a type of mutation involving the insertion or deletion of a nucleotide in which the number of deleted base pairs is not visible by 3. The core reason for the presence of frame shift mutation is the body's mechanism for translating genetic information into amino acid sequences through a triplet-based genetic code. So let's proceed to gene regulation. Gene regulation refers to the mechanism that act to induce or repress the expression of a gene. This includes structural and chemical changes to the genetic material, binding of proteins to specific DNA elements to regulate transcription, or mechanisms that modulate translations of messenger RNA. So this is the importance of gene regulation: to allow cells to adjust with changing conditions, to control the amount of gene product present in the cell, specialization in division of labor among cells, expression of appropriate genes at proper time, synchronized regulation of multiple genes encoding products with interdependent activities, and lastly, prevent the wastage of cellular energy. And that's all. Thank you. And